Hello, and today we're going to be talking about genomics. So, we're going to talk about a few things in this presentation. First of all, what is genomics? Also, why do we study genomics? Then we're going to learn about some strides that have been made in the field, but we will also look at the considerations and limitations that exist in genomics. Let's get started. So, what is genomics? In a nutshell, genomics is the study of a genome, and a genome is all of the information in one's DNA, including both coding and non-coding regions. In this slide, I've also included an image of what we know as the central dogma of DNA. The central dogma of DNA is the replication of DNA, the transcription of DNA into RNA, and the translation of an RNA into a protein. So what is genomics? Before we talk about genomics, let's think about what we know of genetics, the study of how certain traits or conditions are passed from one generation to another. So examples of genetic disorders, also known as inherited disorders, are cystic fibrosis, Huntington's disease, and phenylketonuria. That's a mouthful, isn't it? So while genetics looks at mainly inherited traits and disorders, genomics is a little more broad. Genomics is the study of all of a person's genes, and it also includes interactions of those genes with each other and with a person's environment. So genomics allows us to look at more complex diseases like heart disease, asthma, diabetes, and cancer, which are typically caused more by a combination of genetic and environmental factors than by individual genes. Here's a helpful infographic. Genomics is the study of an organism's complete set of genetic information. The genome is the complete genetic information of an organism and includes both genes and non-coding DNA. Genetics, however, focuses more on heredity and the study of the function and composition of a single gene or genes. A gene is a specific sequence of DNA which codes for a functional molecule. So genomics is a little more broad than genetics. One thing that helped me understand genomics is that the way you live influences how your genome works. While your genome is something that is organized in a certain way, it's still more of a guideline. So genomics ultimately is going to be not only the study of your genes, but how those genes interact with each other and how they're infected by the person's environment. And this makes sense if you think about complex diseases that we can understand a little better with the help of genomics, like heart disease, asthma, diabetes, and cancer, because although you can be at risk for these, how you live ultimately influences how badly you may or may not be affected by these conditions. So why do we study genomics? Well, genomics allows us to better understand complex diseases that may have genetic components but are also affected by the environment. I just said that. So genomics can predict how a person may react to certain treatments or medications, and this can really help because some people may or may not react a certain way to certain medications, and genomics can help us figure out the best method of treatment. Genomics also allows scientists to identify people who may be at risk for certain diseases. So there may be some diseases that have genetic components, and we can see signs of those in the genome, even though symptoms for those diseases may not necessarily be present at that time. Genomics can also help explain human origins and disease risks in regional populations. So genomics can help in a geographic sense. And also, genomics can improve agricultural practices and food supplies. So let's look at a little bit of a timeline regarding genomics. The term genomics was coined in 1986. Since then, the Human Genome Project was undertaken, and they successfully managed to map out the entirety of the human genome. Some successful things that have come from that since are the understanding of single nucleotide polymorphisms. So if we look at this word, it looks really confusing, but if we look at each individual word here, single nucleotide polymorphisms, basically just means single variations in the nucleotides. So A, T, C, and G, that's adenine, thymine, cytosine, and guanine, respectively. These can sometimes vary, and these single nucleotide variations are actually really important because although they seem insignificant, these SNPs or SNPs are often related to the risk of diseases and can tell us about how someone will respond to a medication. So with SNPs, the HapMap was developed, and HapMap is something that is able to measure the SNPs in different populations and regions to figure out how diseases originate and how it may be related to environmental conditions. So the Human Genome was a project. HapMap and GWAS are projects. Single nucleotide polymorphisms is not really its own project, it's just something that we're able to better understand because of genomics. So before I get ahead of myself, back to where I was. GWAS, Genome-Wide Association Study. This is a study that can help determine which SNPs or SNPs co-occur with disease symptoms. So the GWAS is what allows us to figure out how some people may be at risk for certain diseases by looking at people that already have that disease and figuring out how that relates to the SNPs in their genome. So some fields have arisen from genomics, and I'm not going to talk much about these because you'll hear more about them later in other presentations. So epigenomics is the study of DNA modification. Transcriptomics is the study of cellular RNA content. And proteomics is the study of proteins that characterize a particular cell. So considerations and limitations. In the field of genomics, we have a lot of things to consider and some limitations that still exist. So because we're talking about the human genome more often than not, we have to look at the implications of human subjects. There are many ethical limitations and considerations to be made when dealing with human subjects. Gene editing goes hand in hand with this. SNPs allows us to hopefully figure out how someone may react to a medication and possibly develop new drugs. But drug approval takes a very long time, and it can sometimes take decades for a drug to be made available on the market. Something else to consider in genomics is direct-to-consumer testing, such as Ancestry, 23andMe, other programs that allow you to send your DNA in to be annotated and learn genetic information about yourself and relations. So there's a lot of ethical dilemmas and questions that come up with that, especially how your data is stored and what's done with your data after they've collected it. This also generates a very large amount of data, which is difficult to store. These are just some of the considerations and limitations in genomics. In this slide, I've included a short video for you to watch that tells a little bit more about genomics, but will have a lot of the same information. Okay, thank you.